Yes, I'm here.
Can we start now? Can we start now?
Yes.
Okay. Okay. Good morning, ladies and gentlemen. This is Dr. Beleshana, the managing editor of Myco Asia. Today, we are going to have the first of uh, Myco Asia Annual Seminars 2025. This talk will be given by Mr. Izzy from Indonesia. The idea behind the Myco Asia annual seminars is that mycoasians, especially budding mycologists from Myco Asia community, should get a platform to share their learning, share their ideas with the other members of the community. So Izzy has been very active for last 2-3 years with us, as I believe. So he is uh, one of the greatest contributors to Fungal ID group. Today he'll talk about his research from Indonesia. Thank you, Izzy, for accepting our invitation to talk today. And
Thank you also
yeah,
for
you
this opportunity.
You can talk more about yourself first, then you can talk about the research.
First of all, I'm sorry that my English is not so good because it's not s- my native language.
No problem.
So sometimes I got stuck when I want to say something. So my name is Khalid Hafazallah. Actually, everyone can call me Easy. I am from Indonesia, and I was affiliated with Generasi Biologi of Indonesia. So is it start now?
Yeah, you can start now. Yeah, you can share the slides. Yeah.
Okay, let me share the slide. Okay, first of all, good morning, everyone. My name is Izzy, and I am affiliated with Generasi Biologi Indonesia Foundation, which is a non-profit foundation dedicated for spreading biological and ecological education to communities here in Indonesia. So, first of all, I would like to express my sincere gratitude to Miko Asia for hosting this event, and especially to Mr. Belchinoy for the opportunity to share my work here today. So, it's not moving. So, because everyone here is not Indonesian, most of all is not Indonesian. So, I would like to begin by situating Indonesia within our shared regional context, like geographical. Indonesia is located in Southeast Asia with hot and humid and tropical climate. Why is it stop? So, Indonesia is one of the world's most extraordinary biographical mosaics. The archipelago comprises more than 17,000 islands, about 990 of which are permanently inhabited. And these islands from seven major geographical regions. We are recognized globally as one um, mega biodiversity mega biodiversity country. So our border lie two of the world's biodiversity hotspots, 18 WWF or Worldwide Wildlife Fund Global 200 ecoregions, and 24 endemic bird areas. So, Indonesia also hosts about 10% of world's flowering plant species, more than 25,000, like around 30,000 species, with over half of them are endemic. And for animals, we harbor about 12% of world world mammals, sorry, 16% of reptiles, and also there are 35 primate species and nearly about 1,600 bird species. In other words, that our flora fauna is actually well recognized. So we already know about the Wallace line and also the Weber's line, which describe about the transition between the flora and fauna composition in the Sundaland, which is close to Asian composition mainland, and then also to the Sahul, which is where the place is more close to Australia composition, and also the Wallace Sea, which is has the unique composition that transition between the Asia and Australian part. But up until now, we have no idea or still not yet understand about the distribution and also the diversity of the mushroom here in Indonesia. So we already know that many scientists and mycologists say that fungi is around 2.8 until 3.3 million species that hypothesize. But up until now, globally, there are around 15, sorry, 150,000 species to almost 160,000 species described worldwide by Wijayawardene team and Heidi. And also, Hawksworth has hypothesized that the species of macrofungi globally should be around or estimated around 496,000 species worldwide. But globally, we only describe around 27,750 species yet. This based on the description from Pumsakana. But unfortunately, Indonesia has only described about 871. This good as it is if we see in the global scale that we still have less than 5%. But as the mega biodiversity country, it sounds still we have many homework here. So that's why we need a really effort to explore more and also to get the species count more countably. So our data also published that the species concentrated that mushroom exploration or fungi exploration is concentrated in Java. So there are like 658 described from there, and recorded of the remain large islands still under 100, like in Sulawesi, in Maluku, and also in Papua. and also Lesser Sunda Islands. So we still need many exploration activities to record our macrofungal diversity. Moreover, we have stress with the threatened by expanding agricultural. So maybe we will never know if there is or there are some species that exist. Even it can be extinct before we record it. So after that, I got a chance to explore here in Southeast Sulawesi. So let's see. So we did the exploration we conducted by quadrat plots. We conducted 5 plots for each ecosystem, and the plots are 20 times 20 meter. And also we took 9 subplots or in the corner of the plots and also in the center for soil sample. So we try to get the ecological data and also the taxonomical data. So here the map of the Rawa Aupa Watumohai National Park in Southeast Sulawesi. So they have around 6 ecosystems there that we surveyed. So the first one is mangrove, and then savanna, and then we go to the swamp area. So the savanna we conducted into a place. So the left one is the natural savanna, which it has less human activities there, only tourists or some villagers that get around there. But the second one is the disturbed savanna, which is near the road, the the near the road. So we assume and we hypothesize that it should be has different composition of the mushroom as well. So we also move to swamp area. So we make plots surround the swamp at the land that circling the lake. And then we also visited the villagers. So we surveyed in agroforestry systems, which is already influenced by human activities, especially for agriculture. I'm sorry. And also the last part and the most important is in the forest. So this is the method that we conducted. We document and record the macromorphological characters in situ of fungus food body or macrofungi or mushrooms. And then we collected the specimens to the spore prints and examine under microscopy. We also tried to get the sample for DNA in situ. So we just put into the sterile small tubes, microtubes. And also we cultured also in the place that we stay in the field with 15 tube containing of PDA, and the rest of the specimens we dehydrate with food dehydrator. So we collected the soil samples also. We measure the pH, the ORP or oxidation reduction potential, and also the dissolved oxygen. We measure it using a multi-parameter soil device, as I mentioned, that we collected the composite of the soil sample from each plot, from each corners, like nine position from within the plot. And then after that, we identified the fungal taxa based on the macro and micro morphological characters. And if we have or succeeded extracted the DNA, then we analyze it also with the molecular approach. And also at the last, we synthesize all this data and we try to interpret by ecological perspective. So here's the result. So in each ecosystems, so we have 6 here. We have agroforestry, we have mangrove, swamp area, savanna, sidewalk savanna, and also the forest. We found that the family dominated by Polyporaceae. So maybe this related to the decomposition roles of the Polyporaceae, especially for decompose cellulose and lignin. So the highest diversity among the ecosystem is clear, forest, as the bank of the biodiversity. But we still can found mushrooms in mangrove, though maybe we did it in the wrong time, so we didn't find it many. And also we found that in Syro savanna is very tough or difficult to find mushrooms there. So within the family of the macrofungi, even in mangrove and in Syro is less than another, but it doesn't mean that the mushroom is not there. Maybe they just not yet fructify. Uh, fructify. So the beta diversity or the similarity, we use Jacquard similarity index. So we can see that in species level, so the left is the family level and the middle is the general level and the right is species level. So for species level, quite surprising that savanna has a little bit similar with tropical forest. Though agroforestry also has the closer, sure, that's because has a near landscape or close landscape between the agroforestry and forest. So that's why it has same composition of the macrofungal diversity there. So I will show you the different distance of this similarity kind. But here, what I want to show you that based on this heat map, the tropical forest has diverse, and then the sidewalk savanna has no any similar species among with the other ecosystem, and quite surprising also that Riccinellacea is dominate in savanna. So here's for the gamma diversity. So from all ecosystems, we have 82 species that found there and 48 genera and also 30 family. So here's the composition uh, between ecosystems. So if we compare the species, so 53% are located in the forest. And then for the general and also family level, still forest dominated as they are the main biodiversity spot, and also there are so many source there as fungi rely on. Sorry, there is a time calling, so I should pause for Mr. Billy. Can I pause? Thank you for your understanding. Because we have a pair calling. Okay, let we continue. So we have around 53% species that distributed is in the forest area. And then for the composition, the most abundant family level is Polyporaceae. But for general level, We have Satirella that the most abundant there. This may be because of the splitting of the genus. So that's why Satirella are more common there than the Polyporacea. That's it. The Polyporacea that we found in the area dominated by Lentinus and also Ganoderma. So they have multiple role like Ganoderma. They can be saprotroph and also can be uh, pathogen for the trees. And also we found Termatomyces, like around 4 species there. But the most abundant species and the most diverse species in the area are Satirella that we found. So here's the ecological role of each ecosystem. So we found that the ectomycorrhiza only shown in the forest because the host tree available there that dominated by Fagaceae tree species. But there is no ectomycorrhiza species found in other ecosystems. And also for the habitat or substrate. So in agroforestry and forest, there are some of humiculous mushrooms. Mostly, it's almost same like lignicolous mushrooms, but in the forest because there are some of the mycorrhiza occurs there. So the terracolous or the soil mushrooms showed up there. So here's the river of the ecosystem. So the PC1 this is represented by the high ORP and also low pH. So the ORP and pH is usually contradict, while the PC2 is defined by dissolved oxygen of the ecosystem. So in contrary that the forest has a high ORP but lower pH than the mangrove ecosystems. And this may um, predict of the species distribution as well, that uh, ORP and pH may have drive the species composition as well, especially in terms of the soil mushrooms, because in the mangrove, the mushrooms that occur usually is ignicolous. So, brief conclusion from this trial is that one, tropical forest represents the ecosystems with the highest diversity levels, both in terms of alpha and gamma diversity. And then species composition, which is discussed about the beta diversity, varies significantly across ecosystems. Or we can say that we have low Jaccard similarity index or high Jaccard dissimilarity index, with the exception of savanna and tropical forest transition. Citrus savanna and mangrove are the ecosystems that are characterized by the most distinct species compositions. And diversity of all ecosystems is 82 species, which majority belong to family Polyporaceae, up to 17 species, and Satyrellaceae has the highest species among other genera. So that's the biodiversity part. And then after that, I have chance and also another survey in Central Kalimantan. So at the moment, we already collected about 8 species of mushrooms there, and we tried to culture it. And these 8 species, Astroboletus, Trobilomyces, Entoloma, Lesinellum, Calostoma, and also Lactarius was succeeded to culture in our media. And then we try to screen in uh, the activity of Laccase, the activity of Laccase. So we actually just for this presentation, I just want to share the initial screening Laccase production. So it's just to identify if it's positive or negative result of the Laccase activity in ectomycorrhizal strain. So we tried with two indicator, two media indicator. This is conducted with this is cultured by on PDA. We use guaiacol and also guaiacol with additional of copper sulfate, and then we also test with the ABTS screener or and also add the copper uh, s- sulfate. So here we can see that there are positive and negative of the result. Like the M15, we have no sign there that strong reaction. And also for M35 as well, we didn't see any sign of strong reaction. But we have three that strongly positive. That is, they are KH08, M6, and also M12. So what's the mushrooms? So we let's get back to the species photo. So KH8 is the Entoloma species, and then the M6 is Calastoma insigne, and also M12 is Entoloma clypietum complex. So in qualitative assay on PDB, the result is quite interesting because we got the result from M12 very quick. It is less than 10 minutes, like 5 minutes or so. The reaction already showed up. So the liquid culture of PDB or potato Texas growth is already changing. So this indicated that the activity of the Laccase activity by Entoloma pipetum complex is very fast and also has strong initiation. So in the future, so we need to also try to have optimization and also the fermentation time, and also we need to try because we just did the initial steps, so we need to compare between the dark and light environment because some fungi has metabolic influence by light, especially from UV, and also characterization of enzyme properties. Actually, we already done some metabolomics, but the result is not arranged yet. And also the direct use of the strengths of color removal of textile dyes. So maybe it can be potential for Entoloma clypeatum to be the textile dyes color removal. So that's all that I can deliver and share with you. Thank you very much. Sorry if there are many obstacles and also posts. I will give back to Mr. Bele.
Thank you. Izzy, can you hear me, Izzy?
Yes.
Thank you very much. I mean, I'm very glad that I listened to you today. You know, it has enlightened me with what is happening in Indonesia, with respect to mycology and being in other part of the Asia. So it is a good introduction to the geography of Indonesia too. You know, before I ask you, or before the audience ask you the questions on research, I would like to know from you what got you interested in mycology, what got you interested in fungi or mushrooms.
Actually, I like every creatures that got been created. So I love about diversity, everything about ecology that I yeah, everything about this nature is so amazing. So many things that we can get. But why mushrooms? Because here in Indonesia we have a very lack or gap here, especially when we go to identification of species. We have no reference, or I mean, very little reference that we can find out. I was actually before I was plant identification. So at the forest, um, when I found some mushroom species, yeah, I have no idea how to identify it. So that's why I began 10 years ago to learn about this mushroom identification, and that's why I love to share it really to everyone. Um, and also because it's very potential. I mean, fungi has so important and what we say divergent ecological role. They are pathogen, they are mycorrhiza, they connect all of many from life and death. So it's so amazing when discussing about mushrooms.
Can you share, like, how did you learn the taxonomy of fungi, the taxonomy of mushrooms? So, like, what kind of resources you utilized?
So from 10 years ago, when I was when I just as a common forester, so I went to forest quite uh what I usually went to forest because of my work, and then I found some of the mushroom species that edible, and also the some of the villages that eat the mushrooms, but I have no idea about it. And then I consulted first in the Facebook group. So I met Davide Pudu from Italy. So he owned the group, Facebook group, Mushroom Hunter of the World. So from that, I asked him to make Indonesian Mushroom Hunter. Since then, I made many professional mycologists, and also I always discuss with my lecturer. Uh, she's her name is Alice Nina Herliana. She's actually more about the forest pathological specialist. So when I brought some of the mushroom species, she also said that I have limited knowledge about this. So that's why she gave me Germany mushroom book identification. So from there, I discussed with Davide and asked him if he can guide me. Then he guided me, and then in the group, it's so amazing. Many people so supportive, and then I got the chance for professional survey for mushrooms. Then, like in the Sulawesi, so this I learned more about molecular and also how to analyze the genetical data and how to match the morphological mushrooms species into the phylogeny and also analyze that. So that's my journey.
Let me ask you one more question. One of the critical issues in mushroom biology is the edibility. How do you predict the edibility in the field itself, or
Yes.
do you do
Yes.
that?
Yes, of course. Because when I said to when I recommend another person to try on, so it has to be my experience first. But Alhamdulillah, so far I have never get sick by consume any mushrooms.
Can you give me some idea how many edible mushrooms are there in Indonesia? Actually? Any idea?
Uh, in my own documentation is around 110.
Okay, you have
Yeah, my own.
yeah, you have a good idea about 110 edible species of
Yeah,
Indonesia, and you can easily identify them in the field, right?
yeah.
Excellent, excellent. And I will let the audience to ask questions if they have any. Ladies and gentlemen, if you have any questions, please asking. I'm turning on the microphone. Yes. Ladies and gentlemen, if you have any questions, you can just raise your hand and you can ask easy. Anybody has any questions to easy? Okay, they may still be thinking. Let me continue asking some more questions. Now, we had many discussions in our groups. Like, how do traditional morphologists perceive the complexities of phylogeny-based identification? Actually,
For identify the morphology based on morphology.
how do experts of morphotaxonomy perceive the modern complex phylogenetic analysis when it comes to identification of mushrooms? For many species, we don't even have type specimens, and we don't even have sequences or what we call type sequences. So somebody who has spent lots of time in the field, what do you think about those issues?
Yeah, for me, herbarium is the most critical thing here. I mean, unfortunately, here in Indonesia, also our specimens may be unnamed. I mean, we have many collections that still not described in taxa, not yet resolved. So that's why it's very tough for us to exactly say that this is the species. This is the species A. So that's why to species level, I still learn about it. I still learn about it. Even when my research is about Lactarius, I've submitted into as a new species, but from the herbarium, and then we took the DNA, and it's similar. So it's not new species, but it's just new record. My specimen actually already been sequenced by Verpeken from Belgium. So yeah, it's tough. I mean, some of the DNA data is actually already been done, some, but has no name of taxonomical taxa. But in our specimen as well, we have no name in our herbarium. But because Indonesia, fortunately, we had many historical mycologists from Netherlands. That's why we still can little bit follow the taxonomical name and update. But so far, the what we say, the progress for the description of new species is not as rapid as China and Thailand. Thailand is, I think, very fast in.
I would like to know if the audience has any questions. If you have any questions, ladies and gentlemen, please raise your hand and you can unmute yourself and ask. Anyone has any question for Easy? Because many of the people got benefited by knowledge sharing by Easy in Fungal ID group. Many of them recognize you from your work, from your contributions to their journey in mycology. Ladies and gentlemen, if you have any questions to Easy, please raise your hand and unmute yourself and ask. Okay, I have the last question. Do you have many reports of food poisoning and not food poisoning, mushroom poisoning in Indonesia?
Yes, we have two cases of that, but unfortunately, I cannot access the specimen because we have different. I mean, the distance between us is different island. So, but I already recommended to send to the campus or the university there. But the update of the initial identification is not yet done. So we also have in our group in Indonesia Mushroom Hunter, we have 3 cases of poisoning, but it's just sickening. So they got vomit and also and so on. But Alhamdulillah, we can took care from far. I mean, we suggest them what to do, and Alhamdulillah, they got well. Yeah. Hi, Mila.
Mainly, please, you can ask your question. Yeah.
Hi, Khalid. Yeah, I mean, I had two questions. Unfortunately, I wasn't able to make it to the whole talk, but I just had two questions for you. One, you were mentioning the different enzyme reactions with daylight and darkness, and I was wondering if you could elaborate on that. And the second, I was wondering how you're in Malaysia, the engagement with citizen scientists and others interested in fungi has been, yeah, with your team. Also, I apologize, I'll have to turn my mic off. I won't be able to speak, but thank you for your answer.
Sorry for the second.
The second one, I wanted to understand the relationship with citizen scientists and people interested in fungi with your team. Do you ever like have walks, or is there kind of any engagement that you've had with people outside of your team as well?
Okay, thank you, Mila. So the first one, the reaction of enzyme. So IBTS and guaiacol is common for screening the Laccase activities. So it's for initial step, every changing colors. Usually for guaiacol will be red or something like black, and then for the IBTS will change to blue. So that's how the color of the chemical agent for screening the Laccase activities. And the second one, yes, Alhamdulillah, I have engaged with some of the mycologist enthusiasts. Though maybe many of them we still not yet meet up. I mean, direct meet up, but we have intense communication by WhatsApp or by social media. And also, I joined Poison Help. So I learned also how to face the case of the poisoning case when it reported to us. So that's how I learn and engage with some of other mycologists. And at the moment, I am waiting for another research program. Actually, from Singapore. We are going to try apply mycosilviculture. So we try to cultivate ectomycorrhiza like in Italy and Europe actually already done it, but here in Asia we still not yet. It's still just villages that hunting the mushrooms, but not yet in the. We want to establish it in restoration program, so forest restoration program, so we can enhance for community also, for economic also, and also for the ecosystems. And the homework is because we are trying to conduct it in pit ecosystem, so that's a little bit tough to restore. So I'm trying to establish it there.
Very cool. I've actually been very curious about peatland ecosystems, peat swamps, and how the fungal biodiversity is there. So
naturally there are so many swamp. you can suddenly fell into the. But right now, some of the peatland already been human interference, so it will be safe, inshallah.
Thank you for your answers.
Hope you can come here.
Yeah, I would love to. Thank you very much.
Easy. I mean, I think this could be the last question for you for today's session. I mean, if someone is starting his journey in the mushroom biology or mushroom. What would be your some suggestions?
Suggestions?
If someone is starting his journey in mushroom identification or mushroom biology, I mean, you started 10 years back, so you have learned a lot during those 10 years. So if you are to give advice to some young people who are just starting their journey in mushroom identification, mushroom exploration, what would be your advice to such individuals?
So my 50 cent, well, my 50 cent, my golden words is Binokia. So I mean, Binokia is connect with people. So as long mushroom is from spore and then to mycelia, and then they communicate with trees and many of plants. So connect with people. I think there is no use when we have knowledge and then we can give benefit to others. So try to not engage, motivate yourself to be useful and give many benefit to others. So then it will be happen miraculously.
Thank you, Izzy. It has been wonderful. I'm very glad that I could hear you talk today, and I'm very happy that we could connect face to face in terms of video. I think we'll have more talks on the Micro Asia annual seminars. Whether there would be only 2 people or more 500 people, we'll still continue this seminar series because the knowledge sharing is quite addictive.
Yes.
New things we learn are going to make us very excited about the science. So thank you very much, Izzy, and I hope you have a great day. Please continue to stay with us for the second talk on lichens. All right. Thank you very much.
Thank you.
Thank you. Now, ladies and gentlemen, thank you for joining for the second talk. We have Dr. N. Rajaprabhu. Dr. Rajaprabhu, can you hear me? Okay, give me some minutes. I will talk to him and get back to you. Thank you. Ladies and gentlemen, I had just spoken to the speaker, the second speaker. He said he will be able to start in next 10 minutes. So we'll take a break for next 10 minutes, and we'll come back at 11:10 Indian Standard Time. Thank you.